Hello, everyone, and welcome to our weekly webinar. We're giving everyone a few seconds to join, and we can go to the next slide. So this is our weekly webinar, October 12th, 2023, and we were not here last week because we were at a NEALS conference. It was really a great conference with many people from the ALS community coming together, and we presented an update on the platform trial, and so that's what we're going to do tonight for you as well. We're going to present some updates on where we are. As you know, this is a large-scale collaborative effort with many partners in the community listed here, and we really are here to do trials in a different way, in a way that's more efficient, faster, with the goal of getting answers to patients as quickly as possible. And I do want to say that we also have a lot of scientific goals. In addition to testing drugs, we're testing new biomarkers, and the plan is to really share the results with the community as soon as possible. Next slide. So right now, as you know, so far, we have been testing 7 drugs. The testing for the first few is complete, and we are actively enrolling for regimens F and G. When we say regimen, we mean active drug and matching placebo. And so we're testing 2 drugs at this point, and we're enrolling new participants for 2 drugs, F and G. The first regimen is done in partnership with Calico, and again, we are enrolling across the country at several sites. And the second regimen is done in partnership with Denali, also enrolling at several sites across the country. Next slide. And as always, we love to give you an enrollment update, and this was asked by the patient community. They, and for good reasons, people want to know where we are with enrollment. And so I'm pleased to report that we are over 50% enrolled when you look at the current regimens as a whole. So we have randomized 272 participants within F or G. And so what that means is that 272 people have started to receive study drug, which is either active drug or placebo. And as a reminder, for those of you who are new, the ratio of active drug to placebo is very favorable in the platform trial compared to other standalone trials. And the reason for that is that we are sharing a common infrastructure and placebo data across regimens, and that allows us to minimize the number of people on placebo. Unfortunately, we still have people on placebo, but we really try to do everything we could to lower the number to the possible minimal number. And so the ratio is actually 3-to-1. So for every 4 people who enter the trial, 3 will receive active drug and 1 will receive placebo. And that's true for the first 6 months of the trial. But at the end of the 6-month participation, all people who complete the 6 months are then eligible to receive the active drug, even those who were previously on placebo, as part of the active treatment extension. So that's where we are with the numbers. For those of you who are new, if you're wondering why there's more people at the top of the slide, why do we have 382 people who have consented and only 272 who have been randomized? That's because the participation goes through stages. Like in any trial, there is a screening process where people, first of all, they're told as part of the informed consent process, they are, they are informed about what participation in the trial would entail. And there is a screening process to look at characteristics of the participant and safety labs to make sure that the trial is a good fit for that particular person. And so as part of the screening process, again, there is a 6-week period. We try to make it as fast as possible. But people have to flow from basically, the informed consent to the time when they have the baseline visit, which is when they start receiving active drug. So again, 272 randomized, many more in screening, and hopefully most of them will be able to be included over the next few weeks. Next slide. And we are enrolling at 63 sites across the US. That's the map. So we're very happy. Every year, every week, we actually add sites. And now we are pretty much where we wanted to be in terms of having the trial available at least 60 sites. That was the goal. We have a few more sites that we are working with. We're really hoping to open a few more sites that may have been delayed due to various administrative reasons, whether it's staff availability or local permits, local institutional processes. But hopefully we'll be able to get a few more on board. But again, our goal was to have at least 60 active, and we certainly have surpassed that. So hoping to get closer to 70 over the next few weeks. Next slide. If you're wondering, is there a trial site near me, please check our website. We had the QR code, we had the link here, and if you go to our website, we list the sites by state, and we also have the trial contact information. So you can contact the study team directly. We have the information on our website if you click on the name of the contact person, and you can reach out directly. And I see that Catherine is posting some very helpful links in the chat, so feel free to use the chat to go to the link and open up the web page. And as always, you can also contact Catherine outside of this webinar if you have trouble connecting with the site. Next slide. So we are doing a lot of research sort of as part of this trial, not only to test if drugs are effective or not, but also to evaluate emerging biomarkers and outcome measures. And that's very important. That's important because we need to learn more about ALS. We need to better understand the role of biomarkers. And in particular, as you know, there's been a lot of interest in neurofilaments with the accelerated approval of tofersen for people with SOD1 ALS, which was facilitated by the availability of neurofilament data. So neurofilaments or other biomarkers are markers that can tell us something about the disease. And so if, for example, in the case of tofersen, because the drug was shown to affect neurofilament levels, that's what led to accelerated approval in addition to other data. So we really are, doing a lot of analysis to add to our trial, and so we are testing neurofilaments on everyone. We're doing genetic testing, and we are also collecting a number of other biofluid biomarkers as well as digital biomarkers. And by digital, I mean data that can be collected via a smartphone, with the goal of learning more about ALS and also helping the drug development process. I mean, it would be wonderful to show positive effects on neurofilaments, for example, or other biomarkers, and that would help us have more evidence to support new drugs. Next slide. So there's a lot of discussion in the community about biomarkers, so we wanted to bring to you some helpful resources. So the brochure that's on the slide is a new resource that we put together with input from many to explain the biomarkers that we are testing in the platform trial, and also additional resources. So all of this is available for download. You can, again, print them out. You can take a look at them digitally. So there's a variety of resources. And I think, Catherine, I believe that these slides are also posted on our website, correct? So I think, if you go on our website, following the helpful links that Catherine is posting in the chat right now, you can download all of these resources. And thank you, Catherine and Alison, for all your work on these resources and for sharing them with the patient community. We hope that also you will repost all of these. If you want to repost links on your social media platforms, please feel free to do so. These resources are approved by our IRB, which is the Institutional Review Board. They can be widely disseminated, and we really want to be a source of information that might be helpful for people in the community. So even if you know people who cannot join this webinar, please share the resources. Next slide. And we have more resources. Actually, we have a number of brochures and videos. Specifically, I think it's important to focus on the regimens F and G. These are drugs with fantastic science supporting their development. And so there's a lot of information in the previously recorded webinars. We had the scientists from these companies come on this webinar. Everything was recorded and viewable, and they explained really the science in detail. critical, and we will have them back in the fall. I believe that Catherine is scheduling the date for them, so we'll make sure to circulate the dates, but they'll be back. So if you read the brochures or watch the videos and have questions about these drugs and the science, you will have another opportunity to ask questions live on this webinar. Next slide. And as always, again, we're happy to talk to you and connect with you outside of this trial. You can reach out by phone, email. We continue to bring the webinars, and also we have the ALS link, which is our newsletter. So feel free to sign up for that. Next week, actually, I'll be traveling to a conference. Dr. Sukovic will be here. And then we are tentatively scheduling the Regimen G Science webinar for November the 2nd. So we'll confirm that next week and also confirm the dates for Regimen F, which is great. So, so again, and with that, as always, we keep these updates brief because the main purpose of these webinars is to have a live forum for people from wherever. We have people from the US and also international attendees that join us live and ask us questions, and that's really our favorite part of the webinar. So I think with that, we can take down the slides and start taking questions. So the first question is, what does, what's the role of TDP-43 in people with ALS? So specifically, is it that that's normal, abnormal, or that we cannot clearly what's the purpose of TDP-43? So this is a great question. And in fact, I'm glad that you brought up TDP-43. And I think, Catherine, that when we had Dr. Maragakis, he showed a number of graphics about TDP-43. I think he called TDP-43 the guardian of the galaxy, if I remember that correctly. That stuck with me. It's a great analogy. So TDP-43 is like the guardian of the galaxy in the ALS world, in the sense that it is really a crucial protein, a crucial molecule. So in the vast majority of people with ALS, to be precise, 97% of people with ALS, TDP-43 is a critical node. And that's true for pretty much everyone with ALS, with the exception of people with SOD1 or FAS mutations. For some reason, for people with those rare mutations, TDP-43 doesn't seem to be as important. But for the majority of people, 97%, TDP-43 is critical to ALS because it accumulates. And it goes to the wrong location and it accumulates. Normally, TDP-43 is in the nucleus, which is the central core or the central part of every cell, of every nerve cells or motor neuron. And then for reasons that we don't fully understand, in ALS, TDP-43 goes out of the nucleus, goes into the cytoplasm, which is the sort of the general area, the bigger part of the cell, and accumulates there. And cells cannot get rid of it. And so by accumulating there, they actually have a cascade effect. So these proteins that accumulate, they have negative consequences on other things. So that's the problem. So it's not just TDP-43 itself, it's the domino effect that happens as a result of the fact that TDP-43 is in the wrong place and there's like too much of it and it's not the right form. So basically it's not supposed to be there, and by being there, it will essentially initiate a cascade of events. That's why the brand new biomarkers, and maybe we can also post some links to those biomarker webinars, the new biomarkers that we are testing for regimens F and G are brand new biomarkers that are being developed as we speak, or have been developed, and now they're kind of being refined to be able to measure TDP-43 consequences. So in other words, not just the protein itself, but what happens when TDP-43 is in the wrong spot and doesn't function properly. So that's why we're so excited about the new biomarkers that we're measuring, and most of them are only available in the spinal fluid or CSF. That's why it's so important that we get spinal fluid, because that's the closest part of our body to the brain. And that's why we can measure these brand new biomarkers related, or they're basically downstream consequences of TDP-43 accumulation. So sorry for a long-winded question, but TDP-43 is always a great question because it has so many aspects. There's so many aspects to it. So the other question is there a sort of a anatomic donation program? And I will say that we don't have a formal program as part of this trial. I would suggest that you talk to your physician, and it's actually a great question. We know that some people with ALS decide to make very generous anatomical gifts, which means donating their body to research for when they pass. And that's not connected to the study, to the trial necessarily, but there are programs at our hospital, for example, we have a program, and other hospitals in the country do have it, where basically they accept those donations. These are incredibly precious, and they're put to good use for research. So thank you for mentioning that. So I see a question about a new study that's not related to the platform trial, but we are in the Boston area, and we work closely with Spalding Hospital in Boston. And so there is a new study that's run by a colleague of mine using electrodes, magnetic stimulation for testing whether that can be helpful in ALS. And I will say absolutely, if you are interested in participating, we can get you the link. Email us or email. I know you're seeing it at our clinic, so your nurse can tell you more and give you the link, or I can give you the link as well. It's a new study. It's an early phase study, so we really don't know what the results will be. But I think that it's really an interesting study, and it's not a drug, it's a device, and so definitely we need to learn more about the role of devices for ALS. So a few questions. There's a couple of questions actually, so I'll take them both at the same time. So the question is, when do we anticipate the top line results for regimen E? So the top line results are expected within the next few weeks. We always said sort of in the fall, winter, kind of by the end of the year. So we're still on target, and so I cannot give you an exact date because we are still kind of working through the details, but that's what we're anticipating. Now, there's a great question about sort of, recent announcements and the NEALS conference. So I want to take both. So as I said, last week, we didn't have the webinar because we were at the NEALS conference. And that was an important week for ALS research for many reasons. So first of all, at the NEALS conference, it was great to come together and see how many trials there are. Now, I know we take it for granted right now, but I can tell you that just before the pandemic, maybe there were like 2 or 3 trials in the US enrolling for ALS, and now there's like so many that it's almost hard to choose which trial to go in, and many centers offer multiple, so it's really a transformation. So there was a number of trial updates, no results yet again, but we lots of research, and obviously when you have more trials going on at the same time, obviously more chances of seeing something helpful. The other thing, and I want to connect it to the other question, which is a question about some of the recent announcements about NIH grants and funds from the Act for ALS. So those were also announced. Actually, it was a pure coincidence. So the NIH announcements went out during the conference, and that's because the NIH fiscal year ends September 30th. So they announced them basically at the end of the fiscal year, which happened to be the same week of the conference this year. And so last week during the conference, we also had, really we were really delighted to hear about all these announcements. And the reason is, I'm going to talk about them specifically, but I just want to make an overarching comment first, that there's a lot, there's, in addition to the trials, there's many more projects that are going on right now that were discussed at the conference. Some of them are the NIH grants to fund EAPs, but there's more than that. There's other grants or other projects that are highly collaborative. For example, there is a big natural history study that's funded also by the NIH. And there's other studies that were discussed. Some of them are going on in Europe, but it's all kind of good news in the sense that there are big projects that are starting right now. that can really bring about really transformative changes, and we expect them to start soon. So for the EAPs, I can tell you, I personally, I'm involved with some of the expanded access announcements and grants, and we are working on them. So there is generally a 6-month startup period for every grant, for every project really, and we're working on it. So we started working right away, and so we expect to be within timelines, just like last year. In terms of specifically when obviously there's a great follow-up question, so we're starting to work on these expanded access protocols, but when can somebody enroll in them? So first of all, some of these projects are actually already up and running. For example, the CNM AU8 EAP is already available. And now with the grant, it's going to be bigger, but it's already available. We have a few other EAPs at MGH. Some of them are enrolling, some are not. We expect to have the predopidine EAP up and running again within 6 months for sure. We have a smaller predopidine EAP that's ongoing right now, and now we're going to add many more. Many more slots thanks to the NIH funding. So the idea here is to create what Sandy used to call seats on the bus, right? How many seats on the bus there are. So before it was few, and now these grants will allow us to really have many more opportunities for people. There's a question about, again, in terms of the regimens FNG, and kind of it goes back to my previous comments about TDP-43. So the question is, do regimens FNG target TDP-43? And yes, that's why we're so excited about the science. And so again, please feel free to watch the webinars that were done before and come back with questions to the scientists. So these two drugs, address TDP-43. There's other drugs that are, focusing on the pathway because it seems to be so central to ALS. And so that's why we're excited to work on this. Great question about biomarkers. Do ALS clinics track biomarkers? Well, I would say that, in the context of clinical care right now, we can actually measure neurofilament levels. It became commercially available last year, I believe. But it's not really a biomarker study. It's more, in the context of ALS clinics, you can measure certain things that could be considered biomarkers like neurofilaments. What's helpful to research and to advance ALS research is to be part of biomarker projects where the data is collected using standardized processes so that we can actually learn from the data. So it's not just individual level, but it's more collective knowledge from the study. And that can be a biomarker study, natural history study, or a trial. So great question. Can you participate in more than one EAP at a time? Normally not, because these drugs are still experimental. And so just like it's not possible to participate in more than one clinical trial at a time, the same will be true for EAPs. So there's a person who sent a few questions about regimen F drug, how to take it. So I would ask if you don't mind letting us know which site you are from, because again, we just want to make sure that the study team is providing you all the right information. So the drug comes like, small pieces that basically are then mixed with applesauce and so that again there and then with the so that when you take the drug by mouth, you'll take it with applesauce, and when somebody takes it via the feeding tube, it's mixed with water. The drug is basically it's optimized depending on the route of administration, and we don't want to have anything left behind. So the best way to take it when you actually swallow it by mouth is to mix the small pieces of it with applesauce and swallow them. But it's hard to put applesauce into the feeding tube. It actually clogs the tube. So you can just mix it with water, just like you would normally do with any other drug that goes into the feeding tube. So it's not that one medium is not is good or bad. It's just that it's how people take the drug depends on the route of administration. I don't know if that makes sense, but it's basically optimized to make sure that there's nothing left behind, nothing gets clogged or is left as a residue in a cup or things like that. So great question about other regimens. Yes, we're working with other regimen partners. So again, right now we are focused on completing regimen E and enrolling for F and G, but we're certainly talking to a couple of other partners for next year. So more to come on that. As always, it's a combination of things, drug availability and results from previous studies that kind of, we want to be super efficient. So we want to move from, let's say, phase 1 study or an early phase study right away into the platform trial. And so I feel like, sometimes, these timelines shift because there are other things going on, drug manufacturing and things like that, but hopefully they will start soon. So there's a question about sort of, how to choose between different programs. So what I would say is that, for people who are in the platform trial, let's say that somebody is in the randomized control phase of the platform trial, and then there is the ATE or active treatment extension, which used to be called OLE, open label extension, but it's the same concept. Basically, after the placebo-controlled trial, people can take active drug long-term. That's really an integral part of the study. So it's really something that helps us understand if the drug works or not. And this was actually helpful in the recent approvals of tofersen and Relivio. This study design, it's the same concept. So I would say that, personally, I think that if somebody has been in the placebo-controlled trial, it makes sense to stay in the active treatment extension, because then you can receive the active drug. And so, while the ratio active to placebo is favorable, it's still possible that somebody was on placebo during the randomized control trial. So I think that sort of, being in the active treatment extension makes total sense to me. Because again, if somebody is, interested in the particular drug, it just makes sense to get to the point where you're getting the drug for sure. It's true that there are also, now again, we almost have the opposite problem that we had years ago. Just before the pandemic, we had too few trials. Now we have so many and so many programs. And so now the question is, should I choose something different, right? And I think that that's a very personal decision and something to talk to your investigator about. I will say that from my point of view, because all of these drugs are in development by definition, I have what we call it in kind of clinical trials, we call it clinical equipoise, which means we do not know if one drug is better than the other. That's why we're testing them. It would not be ethical to sort of, test some drugs that we think are good and others that we think are not good, right? So they all have the same science. So I think personally, again, that's what I would say. If somebody is in the randomized control trial, to me, it makes a lot of sense to stay in it and get active drug as part of the active treatment extension. But if somebody is not eligible for the trial, and there's other options nowadays with expanded access, and that's great. I think we took all the questions, and I see, Catherine, that you put so many great, links in the chat. And so with that, I think I took all the questions for today. And I just want to thank everyone for your participation. And I won't be here next week because I'll be traveling, but Dr. Sukovic will be here. So today we got lots of great questions, and feel free to come back next week and ask more. And we always love your participation. Thank you so much. Thank you.